Good morning, one and all. Hope you had a great day yesterday. Let's make today's event successful too. I'm Sri Jahnavi of 5th Pharmacies from Sri Vishnu College of Pharmacy. Sri Vishnu College of Pharmacy is the only registered pharmaceutical research center under the Andhra University. Our faculty members receive grant from AICTE, CISR, DBT, and DST, etc. SVCP is the first pharmacy college in Andhra Pradesh accredited by NAC, and our B Pharmacy course is also accredited by NBA. Now I call upon Mr. Anil Kumar Shagi, the coordinator of Pharmacy program from SVCP, to give a welcome note on today's session.
Hello everyone, this is Anil Kumar. So I think you have enjoyed the previous day session, and I welcome you all to the second day National Webinar of Pharmacy Practice. And today we have our guest speaker, Dr. Sugato Banerjee. So today's topic is on gut microbiota and inflammatory disorders, which is very interesting. So I think all the participants will be benefited and will enjoy today's session. I thank, in this occasion, I thank our speaker, Banerjee sir. So thank you so much, sir, for obliging our request on a short notice and delivering a very interesting topic. So looking forward to the topics.
Thank you.
Forward this to the students to please introduce the guests.
Okay. Thank you very much for the introduction. And today we'll talk about gut microbiota and inflammatory disorders. So let me start sharing the screen.
Sorry, sir, we have small introduction about you. Good morning, everyone. I am T. Leela Madhavi from 5th Pharmacy, Sri Vishnu College of Pharmacy. I feel extremely elated to introduce our guest of honor, Dr. Sugato Banerjee. Dr. Sugato Banerjee is working as an associate professor in Department of Pharmacology and Toxicology at NIPER, Kolkata. Sir completed his master's degree in pharmacology at University of North Dakota School of Medicine and Health Sciences. He pursued Doctor of Philosophy in Pharmacology and Experimental Neuroscience at University of Nebraska Medical Center. He also published 30 publications. Sir done research as postdoctoral research fellow at UC San Diego from 2009 to 2011. He also served as an associate professor at Gupta College of Technological Sciences from March 2011 to Feb 2012. He worked as an assistant professor in Department of Pharmaceutical Sciences at Birla Institute of Technology, MERSA, from 2012 to 2020. From 2020, he is working as an assistant professor, associate professor at NIPER, Kolkata. He also volunteered as counselor in Nebraska AIDS Project from April 2007 to 2009. He has several awards and recognitions for his credits. Now I request Dr. Sugato Banerjee to take over the session. Thank you.
Thank you.
Over to you, sir.
Yeah, so I'm trying to share the screen here.
Now you can share the screen, sir.
Okay, just a second. Okay. Is it visible?
Yes, sir.
Yes, sir.
Okay. So I'll try to make it full screen.
So can
It's
we,
full screen? Yeah.
Nice. Now visible.
Okay. Okay. Thank you very much for the nice introduction, and I thank the authorities for calling me for this talk. I hope the students will get benefited from this particular session. So this is a relatively new area that has emerged in the last maybe 4-5 years, gut microbiota and how it affects health and disease. So in this particular talk, we will discuss more about Yeah. Okay. So in this particular talk, we will more in this particular session, we'll talk more about its role in inflammatory disorders. So as you may know, that there are different types of gut microbiota. So it's a whole new world basically. So you have more than 11,000 or more of bacteria which is living inside your intestine, and it affects various physiological conditions, including various inflammatory disorders, also psychological conditions, as well as some established like diseases like inflammatory bowel disease or colitis. And all of you must have experienced, like when you are on antibiotic, you also have tend to have intestinal disturbance, and all this is because the antibiotic kills some of these beneficial bacteria, and some of the opportunistic flora actually are encouraged in the absence of the beneficial bacteria. So here you can see a list of essential beneficial flora versus opportunistic flora. Like you have lactobacillus, bifidobacteria, which are beneficial in nature, whereas you have opportunistic flora like bacteroids, clostridia, so streptococci. So in the presence of this good bacteria, the level of bad bacteria increases, and that leads to a lot of complications, physiological changes, basically. So there is well-established interaction between your gut bacteria and the immune system. So it has been shown that this gut bacteria may be involved, like you can see here, in the differentiation of various immune cells, like you have your T regulatory cells, Th17 cells, Th1 helper cells. So with the help of these certain cytokines and other interferons, basically your gut bacteria can help in the differentiation, or for example, it may increase the level of your T regulatory cells, which are good for your immune system. But it may also, if you have the higher population of this not so beneficial bacteria, you can increase the level of Th17. This Th17 positive cells, basically, they are involved in a lot of autoimmune diseases. Also, we will discuss more about this. And here you can see there is a close interaction between, this is the epithelium, and this is the gut-associated lymphoid region, where a lot of these immune cells basically are getting differentiated from the dendritic cell, mediated by certain cytokines and interferons. So this is again a very well-established concept now, that obesity and diabetes has been associated with change in the gut flora. So again, this has to do with this particular molecule here. You can see lipopolysaccharide, LPS, which actually is released by certain gram-negative bacteria. So if you have a higher population of gram-negative flora in your intestine, it will ultimately lead to leakiness of this intestinal wall, and that will lead to entry of this lipopolysaccharides, which is basically one of the cell wall constituents of this bacteria. And this causes a lot of it can affect a number of organs. Like you can see here, it may cause inflammation of the liver. It may reduce the insulin sensitivity of various organs like liver, adipose tissue, hypothalamus, muscles, and also it may lead to inflammation in all these specific organs. So you can understand, and all these are associated with the inflammation that you see during obesity. So obesity is associated with systemic inflammation. And one of the primary mechanisms is believed to be through this gut bacterial change in the gut bacterial population. So this is one example again here is how the gut bacteria actually causes allergy. This is regarding the food allergy. And again, this food, if you have a leaky intestinal microbiota, or the balance in the microbiota is altered, which leads to leakiness. So these allergens, these antigens from the food can enter your bloodstream, and actually it may actually activate your B lymphocytes. It may lead to degranulation of mast cells and release of histamine, and that may actually affect your allergic reaction to certain food substances. Again here, so you can see your gut bacteria plays a major role in what kind of food allergy you develop. So this is again related to inflammation or liver inflammation basically. So again this LPS, this molecule comes into picture here. This again can translocate to the liver, and basically it may cause so this here you can see these toll-like receptors. So these toll-like receptors, if you are not familiar with this term, is these are pattern recognition receptors. So whenever there is a foreign substance comes into your body, your innate immune response basically is triggered. And these are the first molecules or the first receptors, your TLRs, which recognize this foreign materials. So it can also it can recognize your new bacterial species. It can recognize viruses. At the same time, it can also recognize LPS, and that actually triggers an inflammatory response. And this ultimately leads to heavy inflammation in the liver with the help of these cytokines, and ultimately may cause liver damage and actually can progress to cancer also sometimes. Like you can see here. So again, this is related to autoimmune disorders. Like I initially explained, like IL-17 plays a major role in a lot of autoimmune disorders. So like multiple sclerosis. So here again, the imbalance in the gut bacteria may lead to this citrullination of autoantigens, which is again, so these are antigen presenting cells, that is your macrophages. So it is presenting the antigen to the CD4 positive T cells. And then there is production of autoantibodies through this helper T cell, activation of B cells, and production of autoantibody. And this is associated with a lot of inflammatory disorders like rheumatoid arthritis, again, is one of the disorders, multiple sclerosis, which has been associated with this. And as you can see here, again, this gut bacteria, the imbalance in the gut bacteria is partly responsible for this pro-inflammatory response versus the anti-inflammatory response, which usually the pro-inflammatory response dominates in case of this immune or autoimmune reactions. So this is the case of HIV, the COVID. So in this COVID scenario, again, it has been proposed that your gut bacteria plays a major role. Again, the mechanism seems to be very similar. So like you can see previously that it activates your macrophages, it reduces your T regulatory cells. So as a result of which there is a huge immune response. So you can probably, all of you already are aware that this huge cytokine storm that happens in a lot of patients affected by COVID, and that is causing most of the lung changes and ultimately to pneumonia. So again, if this balance of the pro versus anti-inflammatory response can be re-established, which a lot of physicians are trying to do with the help of corticosteroids, which actually reduces the pro-inflammatory response. So again, this gut bacteria, if you have a good gut health, or you have a good population of bacteria present in your gut, then it will be triggering an anti-inflammatory response. Versus if you have more of a like harmful bacterial population, then it will lead to this pro-inflammatory response, and it will add on to whatever this virus is doing, as a result of which you have this cytokine storm. So here again, you can see this IL-17, IL-1 beta, IL-6, TNF-alpha. These increases in case of COVID. And this again, if you have a good gut health, there's less likelihood. I'm not telling that it will not happen. There's less likelihood that you will have this pro-inflammatory response. You will be more likely to have this underreactive immune response, which does not increase all these cytokines. So this concept of gut-brain communication, actually, again, this is an evolving area, and it is now more or less established that your gut health can affect the mood of the person. And again, here basically the gut and brain communicates through this vagus nerve. So you can see this in this picture. So basically it starts from your intestine and it can reach your hypothalamus. So through this vagus nerve, basically there is communication between the gut and the brain. And here again, serotonin, which is is the happy hormone, plays a major role. And if you have a very good gut health, it actually affects your serotonin levels. Again, these cytokines are coming into picture because if you are you're not having a good gut environment, it is activating these microglial cells. So these microglial cells, basically, these are macrophages of the brain, and they produce the cytokines in the brain. So production of excess cytokines in the brain will lead to certain neuronal damage, which again is seen in case of various psychological disorders like depression. So there's a very clear link has been established between depression and gut health or beneficial gut bacteria. So again, this is a link between the inflammatory disorders of the brain and how your intestinal bacteria is connected to it. So this is how this communication between the gut and the brain happens. And in certain diseases like Parkinson's disease, you can see here, as well as where there is aggregation of beta amyloid. And it has been shown that, for Alzheimer's disease, and then for Parkinson's disease, there is aggregation of alpha-synuclein. So this...
ভাই আমার বন্ধুটার নাম তার ডাক নাম পুটু, তাকে পুটু বলে বলে পুটু বেবি করে না নামই মনে থাকবে না। আসল নামটা ভুলে গেছি, ফেসবুক সার্চ করে ওগুলো ফ্রেন্ড নাই
participants please mute
কিন্তু আমার নামটা আমার মনে পড়ছে না।
Okay, so in both in Parkinson and Alzheimer's, there is dysregulation of alpha-synuclein and beta amyloid, it has been associated with altered gut microbiota. And so what is believed here is basically, again, there is increased gut barrier permeability, which leads to entry of certain microbes as well as their metabolites. And that may increase the leakiness of the blood-brain barrier. And the leakiness of the blood-brain barrier actually allows, again, certain bacterial metabolites to enter the brain. And this actually has been linked with beta amyloid aggregation in case of Alzheimer's, as well as alpha-synuclein accumulation in case of Parkinson's. And it has been shown that certain, like if you were adding this butyrate producing bacteria which is believed to be beneficial, this actually improves the condition of the disease. So this is a very similar kind of like diagram where you are seeing its link with other neuroinflammatory diseases like Alzheimer's, amyloid lateral sclerosis, ALS, as well as multiple sclerosis. So these are autoimmune diseases, especially multiple sclerosis. And like we discussed, there's dysregulation of T regulatory cells as well as Th17 positive T cells, which so there is increase in Th17 positive cells and reduction in T regulatory cells if the balance between the good and the bad bacteria in your intestine is altered. Again, it has been linked to altered blood-brain barrier permeability, altered intestinal permeability, which is allowing certain cytokines as well as certain like compounds secreted by this bacteria to enter the brain and causing all these inflammatory changes. And inflammation is again a key factor in most of these diseases. And in this case, especially in multiple sclerosis, this activation and regulation by the T regulatory cells and Th17 positive T cells, which is responsible for this autoimmunity. So now how we basically fix this? So you have, so these three terms probably you may be familiarized with the term probiotic, because currently a lot of drugs are there in the market used or sold as probiotics. So basically these probiotics are nothing but lyophilized combinations of beneficial bacteria. So basically you are taking live microorganisms, live beneficial microorganisms, and trying to replenish your intestinal flora. So whereas prebiotics, so this is something you may not be very familiar with, especially students. These are basically compounds or substances. Basically, these are all food supplements. Currently, they are not under drugs. So basically, they enrich your gut environment. So most of these are oligosaccharides or fructooligosaccharides, basically complex carbohydrates, which you take, and that basically produces or rather serves as a food for the intestinal bacteria. So it basically promotes the well-being of the intestinal bacteria, which is already present in your intestine. Whereas for the probiotics, you are taking them from outside, live organisms, lyophilized organisms, trying to replenish your intestinal bacteria with good bacteria. If you increase the number of good bacteria, automatically the bad bacteria reduces. So this is how it works. And the symbiotics are basically a combination of the two, which may act as a prebiotic as well as it also have some live bacteria, for example, soya milk. So this is basically, it acts as a food for the bacteria that you already have, as well as you are taking some more beneficial bacteria from outside. So it's a combination of the two. So, but we don't have too many symbiotic products. There are some which are coming up, but mostly we have a lot of probiotics in the market, and the prebiotics are also there. They're a bit expensive now, but mostly they will also be affordable soon. So these are some of the common probiotics in the Indian market. The Bifilac, which is a very popular one. Also, you have some beneficial yeast, like your Saccharomyces boulardii. So this is again a very common probiotic that is recommended, especially people on antibiotics. So these are regularly recommended by physicians, especially to patients who are antibiotics, because what your antibiotic basically does is it kills, irrespective of the type of bacteria, it will kill every bacteria. So it will kill both the bacteria for which you are taking the antibiotic, as well as it will kill the beneficial bacteria that is present in your intestine. So there is overall depletion, but you don't want it to kill the beneficial bacteria. So physicians recommend probiotics. Also, curd is a very natural probiotic because the presence of lactobacillus. So they may also recommend you taking curd along with antibiotics. So these are some criteria of prebiotics because this is a relatively new area which is emerging. So these are non-digestible carbohydrates, and they should resist or they should not get degraded in the gastric acidity as well as by the enzymes present. They must not be susceptible to fermentation by gut bacteria, and also they should be able to enhance the viability of the beneficial organism. So this is the primary thing. Basically, they are meant to increase the viability and the health of the beneficial organisms that are already there in the system. So these are some of the common prebiotics, and you can see here is a lot of them comes from normal. If you have a healthy diet, then you automatically get a lot of prebiotics. But most of them, as you can see from this next slide, these are oligosaccharides. Chemically, these are basically you can see these sugar moieties, different ordered in different manner and with different numbers of this carbohydrate moieties. So basically, these are complex carbohydrates, which are usually not digestible by enzymes, or they don't degrade in the acidic pH. So they reach your intestine and they undergo fermentation, and that helps in producing the food for this intestinal bacteria. So now we'll go a little bit into the research mode. This is the overall literature or our understanding of this area that we discussed. So now if somebody wants to do a study on research on probiotics or gut bacteria, basically our lab is involved in this kind of research for a while now. So how you can approach this kind of studies? So very common ways of inducing gut dysbiosis in animals is through antibiotics. So here mostly we will discuss on the preclinical studies or preclinical pharmacology of gut bacteria. So basically, most common antibiotics like your ampicillin, azithromycin, chloramphenicol, amoxicillin, these are well known to induce gut dysbiosis, or they will kill the beneficial bacteria. And then Western diet. So we call them Western diet because it is rich in high fat as well as carbohydrates. So processed food basically. So whatever you take, your pizza or your burgers, so what is very popular in the West, they are very good to kill your bacteria. So as you can understand, they are not at all good for your health, because they will also in the long term, they will also kill most of the good bacteria in your intestine and promote the development of non-beneficial bacteria, which may actually ultimately lead to sequelitis or intestinal bowel disease, IBD. So a lot of complications can happen if you for long consumption of Western diet, and one of the reasons for that is you are depleting your gut from the good bacteria. And in the experiments also, you can utilize the same thing. Basically, there are commercially available high-fat, high-carbohydrate diets, which if given to animals, others, and we have also seen it actually depletes the intestinal bacteria. So what people do after that is, after you have depleted the intestinal bacteria, so maybe you want to test some new probiotic through your preclinical experiments. So basically, you can look at the whole gut population. So isolation of bacterial DNA from the fecal samples of these animals, you can go for 16S RNA sequencing, and then you get a total phylogenetic analysis. So what genus, what species of bacteria is actually present after before the use of, say, high-fat diet versus after the use of high-fat diet can be measured. and then you can give your therapy to those animals and see whether that is improving the good bacteria versus and reducing the bad bacteria. So this is some results from our lab. So basically, we were looking at the role of gut bacteria in learning and memory. So here, basically, this is these animals were given ampicillin, sodium, and this data was done using the RNA isolation from the fecal samples, and then estimation, relative quantification. So here we used this yeast, beneficial yeast as Boulardii, as a probiotic. And here you can see there is, after antibiotic treatment, the relative abundance of certain bacterial population like Lactobacillus, Bifidobacterium, Firmicutes, Clostridium, they reduce quite a bit. But after application of this particular beneficial yeast, there is increase in this population. So clearly it shows that whatever you see in clinically, that when you are on antibiotic and you're given some probiotic, basically your intestinal condition improve. The same thing happens in animals also. And like this, you can explore newer probiotics and newer prebiotics and whether they're actually working or not. Again, this is something like related to the intestinal permeability. So it has also been shown, and we have also shown that antibiotic actually makes the intestinal wall, damages the intestinal wall. Here you can see this is a healthy intestine, and here this is after antibiotic. And after prebiotic, it improves to a certain extent, but still it is not like initially. But here it is very striking to see how much damage basically antibiotic can do to your intestinal wall. So again, the message here is, again, don't take antibiotics just like that. Only take whenever it is necessary. Otherwise, you will basically do a lot of harm. Not only with respect to antibiotic resistance, that's a different area, but also your intestine is highly affected where antibiotic treatment. So it's not a good practice. And we saw this in animals also. And after the application of the Boulardii or this particular probiotic that we used here, there was improvement, but it never reached what it was before the application. So again, this is one more assay that people do is the gut permeability assay, where we use FITC dextran. We measure the level of FITC dextran in the plasma, and you see here in this particular figure that after antibiotic treatment, there is increased permeability. Again, this is correlated with increased intestinal permeability, the level of FITC dextran. And again, this improves after this SB is basically this treatment of this beneficial yeast. It goes back to normal. So after this, people can look at various other diseases which have been associated with gut bacterial changes. So we are interested more in the role of gut bacteria in learning and memory, but others have been studying the role of gut bacteria in depression, in arthritis, in cancer, and definitely the big one now is in obesity and diabetes. And it is known to play a major role in all these disorders. So this is some data from our laboratory regarding its effect on learning and memory. Again, this is a very new area, and it is just getting established in the scientific community. So I will not go much into the details of the methods, but basically what you can see from these graphs is antibiotic actually, this studies is step down latency, transfer latency. So if you're not aware of these studies, so the take home here is gut dysbiosis actually reduces memory. And then once you give the probiotic along with the antibiotic, it can replenish it to a large extent. So that is a take home from this whole slide. And we do a series of studies on animals to prove this. Again, this is a Morris-Waterman test, which actually tests the spatial recognition of the animal, how well it can recognize. Basically, here there is a platform, hidden platform we put in the water, and how easily it can recognize the area where the platform is put. And it actually tells you the recognition memory of the animal. And antibiotic actually reduces this spatial memory for the animal. And then once you give the probiotic along with it, it again gets back its normal recognition of the spatial memory. So we also did some histopathology, and probably you are aware that hippocampus is the primary region for learning and memory in both human as well as animals. And we saw here that your antibiotic treatment actually reduces the hippocampal neuronal density, so which are involved in learning and memory. And when you add your probiotic to certain extent, you basically get it back. So here, though we don't get it, like the density does not reaches whatever it was initially, but we get it back to certain extent. So you can understand it also has a huge change in the brain. And so again, don't indiscriminately use antibiotics. And if you are taking antibiotics, always try to take, in this case, probiotic, which will actually reduce the damage. But it's not very clear whether it is completely reversing it or not. It reverses to a certain extent. So these are one product I saw in the net that basically this currently this area is being commercialized heavily, and like how your gut health can actually dictate to a lot of diseases. People are slowly getting aware of that. And this particular company, this Ombre, it is selling this stool collection kits where you can collect your stool samples, and then you can send it to that company, and they will do a whole genome sequencing of what all bacteria is there in your intestine based on your stool samples. And it will also tell you that how this bacteria is related to your health. So whether some bacteria which is known to be related to heart health versus brain health versus liver health. So like that, it will give you some, so not only it will tell which bacteria is more and which bacteria is less, so it will give you an idea that whether you are having a healthy gut or you have more of an unhealthy gut bacterial population. But at the same time, it will also tell how it is related to various diseases, or what diseases you may get in future if you don't rectify your gut population or gut bacterial population. And it will also give you recommendations of what kind of food you should take. So based on whatever bacteria is reducing, how can you improve or how can you increase those particular species or genus of bacteria by taking what food, whether you should take more pulses, where you should take more fibers, whether you should take like certain fruits. So it will give you a whole list. So you can go to their site and you will get all these details. So basically, this is how you can understand it is important to health, and it is heavily commercialized to basically customize your diet, because diet plays a major role in your like what kind of bacteria. If you are new, even if you are not on antibiotics, still what kind of food you are taking will dictate what kind of bacteria you will have in your intestine. Obviously, geographical locations, other things also comes into picture. But if you are not taking those into consideration, at least your food habits as well as taking of antibiotics are very important for an adult gut bacterial population. For children, spread is a bit different. It depends on whether they are consuming mother's milk and or they are taking other sources of milk. So those things comes into consideration. But for adults, it's more of taking of antibiotic as well as what kind of healthy diet you are on. So this is one example where all these recommendations are given based on your stool samples. So again, this is an evolving area. So with respect to clinical pharmacy, so it has been proposed that a time will come based on which bacteria you have in your intestine. It can be predicted whether you are more susceptible to cardiovascular disorders versus less susceptible to cardiovascular disorders. So this picture basically shows you, so collecting the stool sample, then metagenomic analysis, where all the bacterial population, whichever is there in your system, gets analyzed, and then it is told like, okay, fine, this few bacteria are more versus the others. And then through machine learning and other tools, it can be predicted, or it will be predicted in future. This is a futuristic slide that whether it can, in this case, it is showing cardiovascular disorders, but it can may be able to predict some other disorders also, brain disorders, kidney disorders, liver failure. So many things that you may get in future if you don't rectify your intestinal bacterial population. So this is coming. This may come in next five years or so. And so you can understand, so basically your gut bacteria acts as like a footprint of what kind of disease you may be susceptible to. And this is a very simple process. Like correlating is a difficult process, but getting the sample is simple here because it's a non-invasive process. You're getting your stool samples and then analyzing it. So it's a non-invasive process, would be very easy to be implemented in population. The process of consent is not a big issue here because you're not going into any invasive procedure for collecting the samples. So this may happen in future, and probably this is a very good thing to do. Some caveats. So obviously everything is not so good in a particular whatever approach it is. It always have some disadvantages. So this is basically, the slide summarizes the excess use of probiotics. So there are reports that patients on this Saccharomyces more likely to get digestive tract diseases because of like growth of certain other non-so beneficial bacteria. Fungemia, that is basically excess fungal population in the plasma, may happen with long-term use of this Saccharomyces. Development of antibacterial resistance or antimicrobial resistance is again a big issue because it has been proposed, it has been seen in some cases also that resistant genes being transferred by this probiotics, which are beneficial bacteria, but it is transferring the resistant gene from one organism to the other, which may actually lead to antimicrobial resistance, which is a very, very dangerous thing. And also certain studies needs to be done before a probiotic is recommended. And currently, because it comes under nutraceutical category, it is not under heavy regulation of FDA. So it does not come under the Drug and Cosmetic Acts also because it's a nutraceutical. So the toxins and the hemolytic potential of probiotic needs to be tested before giving on human, as well as it needs to be tested whether this particular probiotic is infectious or not before it is recommended to human. So these are some of the caveats or some of the disadvantages that may show up with probiotics. This is more specific to probiotic use. But prebiotics, since they are natural in nature as well as it's part of your food that you are taking, it may be relatively much more safe. But again, time will tell if that also shows some disadvantages. So I think with this, I will end my presentation. I thank my students basically who have been involved in a lot of the work that I showed. And also I'm extremely thankful to the authority for giving me this opportunity to share my thoughts, share my research with the students of Sri Vishnu College of Pharmacy. Thank you very much.
Thank you, sir. Thank you so much for the session, sir. So relatively, this is a very new topic, I think, sir. Age-old practice, but we don't have any scientific evidence, maybe, sir.
Yeah, it's a relatively new topic, and slowly more scientific evidences are coming up, and so everybody is encouraged to work in this area, because a lot of the traditional things that we do, they don't have scientific evidence, and once they have scientific evidence, then the new generation also are more encouraged to follow all these practices. And also the West is slowly realizing this, and they also need scientific evidence. Yeah. Thank you.
Thank you, sir. The session is very enriching, sir. There are a few questions YouTube live, sir. So can I please ask this, sir?
Yeah, sure.
So one question is, they're asking what is the basic equipment we need if we have to do these experiments?
Okay, for the behavioral studies, basically we use a passive avoidance apparatus, which is very simple and easy to get, and also we do novel object recognition test. Basically, again, this is very simple, and any institute can have it. This Morris water maze system is a bit complex, but it needs a tracking device, which is relatively expensive. But other than that, the rest of the studies can be done. And this sequencing, you have to outsource it. So once you collect the intestinal samples or the stool samples, it can be outsourced. There are a lot of basically CROs working, and they will be like analyzing all those samples and give you the results.
Hello.
Jnanachandran, sir.
A very good morning, my dear pharmacy professors and colleagues. This is Professor Jnanachandran, who is basically specialized in allergy medicine management. I am associated with the Bangalore Allergy Center. Namaskar. Very good presentation by our Dr. Sugato Banerjee. Normally, this allergic rhinitis associated with the intestinal flora. There are many chemicals, bacteria, viruses, which causes allergic rhinitis. For example, this oleoresin capsicum, that is called chili powder. If it goes, it causes allergic rhinitis. Like that, there are many environmental polluting agents like antigen presenting cells, and these allergic rhinitis are basically mediated through the immunoglobulin E, IgE mediated allergic rhinitis. So here our discussion is about the probiotic. What is the function of probiotics in recovery? Because allergic rhinitis is one of the symptoms of allergy. Basically, allergic symptoms like allergic rhinitis, itching, swelling, pain, and all these sequences of allergic march is taking place. And probiotics has got a very excellent immunity enhancing capacity, and the immunity enhancing capacity increases the immune tolerance level. Am I audible?
Yes.
Audible, my dear colleague? Huh? Okay.
Yes.
See, now we in our Bangalore Allergy Center, we give a probiotic which is containing lactobacillus, bifidobacterium, and other combinations above 50 billion IU to be taken in boiled and cool water, and then put the probiotic granules, shake it, and activate it, and drink after 5 minutes, so that the lactobacillus will get activated and will increase your immunity power. So like that, our Dr. Sukato told about these nutraceuticals. We have preparing a nutraceutical containing curcumin. Basically, curcumin is anti-inflammatory in chocolate form. in chocolate form. When you keep this curcumin chocolate, we call it J-Lo in our Bangalore Allergy Center. When you keep it in the mandibles, you will not chew it. Keep it in the mandibles, it will dissolve of its own, and the curcumin will get absorbed, and it will give a very powerful anti-inflammatory effect. So, like that, many things are there. Nutraceuticals, as you rightly said, nutraceutical does not come under Drugs and Cosmetics Act, and our pharmacy people, they can make it. Many things which I can tell about this nutraceuticals, allergy, how the allergy comes, how is the management of allergy, immunotherapy we give it. In fact, in our Bangalore Allergy Center, it is not. It is sublingual immunotherapy. If we be permission, the organizers can you present few highlights about this allergy? Hello?
Yeah.
Can they continue?
Yeah, sure.
Okay, but normally this allergy is because of various environmental like pollen grains, chemicals, dust mites, and the insects. So we want to find out what is the cause of the allergen. After identifying, we give the particular allergen, we extract. For example, if it is a dust allergy, we extract the dust, and then we isolate the allergen, and then from the dust we extract the glycoprotein, and that glycoprotein we make it in their sublingual dosage form. They call it sublingual immunotherapy. SILIT. This is the novel approach to enhance the immunity tolerance and treat allergic symptoms.
Yeah, it is very
Okay,
nice
this is
actually. Like even the kids can have it. So
yeah, kids can have it, and children have it. So this is a very novel drug. Normally, we give antihistamines for management of allergy.
yeah, that is not. It has a lot of problems.
Antihistamines has got a symptom, but it has got its own side effects, and moreover, it cannot be given for longer period.
Yes.
So we give a pharmacotherapy initially combined with immunotherapy. As the symptoms reduces, we slowly withdraw the pharmacotherapy and remove the continue the immunotherapy. We call it SILIT, sublingual immunotherapy tablets, which is containing 18 microgram of the allergen. Very minute dose, so there won't be an allergic manifestation because of that allergen. Okay, it is like a vaccine. It is like a vaccine. We give it. So subsequently, the patient recovers. We are practicing since about 4 decades about this. It has undergone a lot of changes. Now we are coming in the oral sublingual immunotherapy tablets to be kept below the tongue, and it will dissolve and it will slowly get absorbed and increase the immunity tolerance, and the allergy is reduced. So like that, many things I can discuss. Just for sharing my experience to the audience, to our faculty members, I came in the line. I thank you the organizers and Sugata Banerjee for a very good presentation. And once again, my best wishes for all my pharmacy professional colleagues to enhance their knowledge and improve their professional status. Thank you very much. Okay, good luck.
Thank you, sir. So one more question, sir. It's, antimicrobial resistance and biota. So we are talking about this. Particularly, and so what are the components they're asking? What are the components particularly involved?
So antimicrobial resistance through probiotic use, it's a big problem. It's a basically evolving problem. So what happens here is basically you have, you take one antibiotic. So a particular population of bacteria gets resistant to it. The same thing may happen here. You are taking the antibiotic, you are giving the probiotic. So this probiotic bacteria becomes resistant of the antibiotic. Okay, which initially may look good because it is not getting affected by the antibiotic, it will live happily. But what happens is some of these probiotic bacteria which is resistant to this particular antibiotic, they may transfer this gene to the bad bacteria. So that material through plasmids, it may go to the not beneficial bacteria which you want to kill with this antibiotic. Now, this bad bacterial population also becomes resistant to that antibiotic. So that is the process, and that is maybe a problem if you are taking probiotic for a long time.
Thank you so much, sir, for the session. So we are really enlightened, sir, and we'll try to at least start simple experiments with this topic, sir.
Yes, definitely. If need any assistance, I am always there. You can contact me. And it's very important you start in these areas which will actually benefit the students also and benefit the society also in the long run. Thank you.
Thank you, sir. Thank you so much.
Hello, Dr. Banerjee and my dear Anil Kumar Saki. Antimicrobial resistance is very common all over the world. So normally we have to go by the basic antibiotics. It is there in the WHO standards in all the Ministry of Health. When I worked in abroad in MOH, Ministry of Health in Kingdom of Saudi Arabia, in primary healthcare centers, basically we give amoxicillin, penicillin, tetracycline, erythromycin. With that, we can cure 80% of the diseases. Antibiotic, and there is antibiotic stewardship, how to go ahead, and there is a lot of things which we can discuss about the antibiotic medication administration, how the resistance develops, the antibiotic frequency, dosage, therapy, all those things we can discuss, because amoxicillin, what is the dose, how long to be given. Normally in our society, in Indian situation, nobody is giving antibiotic in a proper dosage and proper sequence. So that is one of the reasons we develop this antimicrobial resistance, and particularly for the tuberculosis, which is a long-term therapy, the antibiotic has to be given in a periodical dosage and correct dosage and for a correct period of about 6 months. So that is why it develops multi-drug resistance. So these are the some of the my opinion for the benefit of our pharmacy professional colleagues, which will be can take it as a take-home message. Antibiotic normally not required. In fact, for example, if there is some home remedies. Garlic that is it is got a very good antibiotic effect. So like that, many things are the home remedies. So this one message I want to convey to my colleagues. And once again, I thank you for the Sri Vishnu College of Pharmacy team and our Sugato Banerjee for listening to my experience. Thank you once again.
Thank you.
Thank you, sir. Over to students to
Thank you.
give their concluding notes.
On behalf of Sri Vishnu College of Pharmacy, I thank Dr. Sugato Banerjee sir for this informative session and also for nurturing our students and making it a grand success. And I thank everyone for attending this event. And the respective feedback of today's session and the session, tomorrow's session will be sent to you soon. Thank you, everyone.
Thank you.
Thank you. Thank you. We'll close the session.